Okay. Make sure here. Okay, Jemiah and Kenna, if you're able to see this, can you guys see the screen okay? Just toss a thumbs up, that should come out. Excellent, thank you, Jemiah. Okay, so let's go ahead and get ourselves started. So, chapter 1. Oh yeah, Kenna, 1117 has been flooded out. That's why we had to relocate to 1019. So it's kind of down the hall from the butterfly on your right, if you're coming west. I get so turned around in this building, but anyway. Let's go ahead and start taking a look at biology. So a couple of things that I want to kind of lay down for you guys is kind of getting kind of our sort of philosophical setting down, right? There's a lot of different things in biology, like concepts in biology that will drive biology. And I mean, there's a lot of good ideas in it, right? So there's a lot of different ways to kind of approach it. I mean, every instructor kind of has a kind of a nuanced sense of kind of their philosophical approach to it. One thing that I always like to sort of be upfront about my sort of philosophical approach and things like that, which is essentially very easy. It's very simple. I tend to be a very kind of a scientific purist. And I strive for, I don't always get there, right? But for intellectual purism. Kind of? Yes. Yes. You're good. So kind of like so I kind of see them as kind of, there's different ways of thinking, and we'll talk a little bit more about this later on in chapter 1, because that's kind of what lays some of the underpinnings of how science works. And a lot of times understanding science kind of requires how it works exactly. But I'm a bit of a scientific purist. Okay, so I think science is an awesome process. I think it has a lot to offer. It's exceptionally powerful, but it's also very ineffective when it's not used properly. Okay, and I see a lot of improper use of it. There's really two things that, in terms of my purist background, my purist sentiments, that I really feel is very, very critical. And I feel like I would be in breach of trust, actually, if I didn't accentuate these all semester long, actually, right? Because part of the thing is, to understand science is you kind of have to understand how to think scientifically. And it's a bit of a different way of thinking than like what you're trained to do when you're coming up through the humanities or through lit or things like that, right? It's a completely different way of thinking. But in a lot of ways, you kind of have to learn how to think scientifically before it kind of all starts to, okay, that starts to make sense, right? And I'm very honest about it, right? We have different ways of thinking, and I'm not saying that one is better than the other. They're designed for different functions, okay? And so the reason why I'm a purist is because I like to try to outline science and the way we think about things in science and biology specifically, so that you can understand with better clarity how science works, how you think about science, and how you don't think about science, right? The less effective ways about poking the bear when it comes to this. Now, when I say that there's a lot of great ideas in biology, I think of biology in particular as like this beautiful tapestry of life. And if you've ever looked at a tapestry up close, a tapestry isn't the function of any one thread. rather a tapestry. The effect of a tapestry is basically the accumulation of many, many threads kind of summing up their effects overall, okay? And so that means that basically when it all works well, no one thread is better than another. And there is not a dominant thread, right? Because they all have to work together in balance in order to make the entire effect. And the reason why I say that is because the ideas and the concepts that we have in biology are very similar to the threads of a tapestry. They all have slightly different things that they do, slightly different effects, but they all weave together. And when well balanced as they weave together, they create this really amazing tapestry of life. Now, when one of those ideas gets out of balance, I am not shy to poke at that, because we do ourselves no favors if we get ourselves out of balance. Okay. The other thing that I always look at in terms of purism is that in science, everything has to be testable, right? I mean, that is a linchpin of science. It has to be testable. That's what sets science apart from, say, philosophy and theology. We'll talk a little bit more about that later. Right? It has to be testable, and you have to be objective, right? Because the one thing that really sets science thinkers apart from other thinkers is a scientist is not supposed to fall in love with their own idea, right? You're not supposed to do that, because the moment you fall in love with your idea, you cease to be a scientist. You're not thinking objectively, and you basically become an advocate, not a scientist, right? Because the harsh metric of science is you may think you have it right, but you need to be objective, maintain your objectivity, because if somebody does an experiment and they say, listen, what you think is right is inconsistent with what I just saw experimentally. So you have to be willing to say, listen, you know what? I got it all wrong, or I got it kind of right, but it's certainly not the way I thought. So you have to be willing to swallow your ego. And you can only do that if you stay objective. When I see areas of biology that are starting to get out of balance in that way, I poke the bear. So you will hear me, when I say there's strong points in biology, I'm gonna say these are super strong. When there's some softer spots, I'm like, you know what? These are a little harder. We're still grappling with these, but I'm gonna be honest, we're not sure, right? So admit where you're not, admit where you are, because that's the only way you're going to grow. I'm a big proponent of that. Now, the reason why I say that on this slide is because one of the biggest threads that are obsessed about and that are way out of balance is this one right here. And I say that as having gotten an evolution and ecology degree at UC Davis. So that is kind of surprising, but when you look at it, you look at all the rest of it, and it's like, we need to be able to modify the way we see this. Why? Because the way we see this, and especially in conjunction with this guy here, right? I mean, certainly who deserves all the credit for it? I mean, I'm not saying don't give him credit for it, but largely most people have an antiquated 19th century paleontological view of evolution. I would argue that evolution is in dire need of updating. The problem is, how do you update that without being castigated as a heretic? It's like, look at the data, right? Stop falling in love with your own idea and be a scientist. I have a lot of strong pokes for that particular bear. So in cases where evolution is really, really cool, right? Because it is probably, of all the ideas in biology, one of the biggest. I mean, it encompasses all sorts of disciplines. It is rangy, it is deep, it is multifaceted, and it is fascinating. And there are some of those facets that have extremely strong explanatory power, right? Which is your ability to explain natural phenomena as it exists. And we have no problem with that part, right? And then there's areas of the theory where it's a little, it's like, well, we don't really know for sure. This is a little harder to wrap our heads around. And they still need a lot of work, and we're still working on them. And we need to be honest about those. So where there are areas where it's like, this is not as well understood, textbooks always write themselves as like, we know that this is true. Like, well, not really. Actually, if you're going to get back in there, there's a lot of competing ideas, which pretty much means that you're not certain exactly how it all went down, but you've got multiple ideas at how it might have went down. That's not really intellectually honest. In a lot of ways, I think for the sake of simplification, oversimplification, I would say, and brevity, a lot of textbooks straight out lie to you. And they make this idea seem easier than it actually is. Matter of fact, the reason why it's so fascinating is because it's complicated. And there's a lot of answers we're still looking for. And that's a good thing. That's a good thing, right? So I always say that, right? So if I need to poke the bear, I have no problem poking the bear. So just remember that. And I'll talk more about that, right? So the important thing is in biology, what we see a lot of is diversity, right? I mean, if you go to a zoo, I mean, just go to Denver. It's not a great zoo, but it's all right. It's got animals, right? But I mean, if you go to a zoo, I mean, you'll see the diversity there, right? I mean, we all have our favorites, right? Like giraffes or whatever, lions, things that we always like. But then you'll see other things that's like, whoa, what is that, right? You know, every now and then you'll kind of be surprised a little bit, not at the Denver Zoo, but in a good zoo, you'll be kind of surprised. I'm picking up the Denver Zoo. They're all right. They're all right. They're not great, but they're okay. As long as my kids like it, I'm good. I'm good. So I feel a little sad for some of the animals there, but it's like, but that's a general zoo thing for me, not anything against Denver. Right. But so the question is then, when you take a look at living organisms out there, you got a lot of them. And do they all look the same? No, they look dramatically different, right? So there's a lot of stuff out there and it all looks different. That's diversity. And so one of the things in biology we noticed is that this diversity is like overwhelming, right? Especially when you take a look at when we first started kind of wrapping our heads around diversity. I mean, for the first time, actually, and Darwin's actually a perfect example because Darwin came around in the early 1800s, right? That's when he kind of took his famous around the world trip on the Beagle. And he largely kind of collected stuff, right? So he kind of like, he was a natural historian, which is kind of what you called a zoologist back in those days. And basically what a natural historian did was they kind of went around the world and they kind of collected stuff. They did animal watching and they kind of brought it back, right? So, I mean, kind of what Darwin did and what most natural historians did, like James Audubon with birds and so forth, is you'd kind of go out somewhere, you'd find some sort of crazy animal you've never seen before, kill it, which we try not to do now, but that's what they did. By the way, if you've ever seen a good Audubon picture, you guys like Audubon pictures, his picture of the birds? Have you noticed how they're all like twisted and contorted like that? That's because they're dead, because they're just like a dead body flopped on the table. That's why they all look weird in Audubon's pictures, is because they're all dead when he painted them. It wasn't until much later that we actually, changed that a little bit. But that's just the way they did things. I mean, I'm telling you, 19th century, we were not elegant at all. We thought we were, but we were not. And so what he did basically is he collected all these organisms like every natural historian did, or make notes on what he saw, and then he would bring them all back. And so here he is in England with all this stuff, all this diversity, all these different creatures, and then he's trying to make sense of it all, right? Now, the thing is, though, when we see all of this, and this is kind of like the first time that we really started to wrap our heads around what we had out there. I mean, up until the 19th century, when these natural historians started kicking in, we really didn't know, like, a lot of the animals out there. Matter of fact, you probably are well aware of most animals on the planet, or at least a lot of them. You've seen pictures of them maybe on TV or National Geographic or just growing up, right? You know they're there, even if you haven't seen them in person. But in the 19th century, they didn't even know these things existed, like the aye-aye, for instance. I mean, here's one, right? Because it's nocturnal, so nobody's in a Madagascar jungle at night, for the most part, in the 19th century, but we are now, right? And so we see aye-ayes, and so if you see an aye-aye, it's like, oh, wow, this is like really, really weird. I've never seen this before. That happened a lot in the 19th century. We really didn't have really much of a clue about how much diversity was there. So we really were, and over the next 100 or so years, I mean, it was just like we were collecting all of these new species, all of these new organisms that we've never seen before. And of course, the same thing always happens, doesn't it? Whenever you have all these things going on, what's the first thing you want to do when you have all this sort of lab full of stuff? You want to organize them, right? That's just a natural thing. So what we did was basically we divided this biological diversity into a system that we invented called taxonomy. And what taxonomy is basically the classification of organisms by morphology. So literally, you do what you always do when you organize and classify a bunch of stuff. Okay, all you cat-looking animals, you go over there in that pile. I'll sort you guys out later. All you dog-like things, you gotta go over there. I'm gonna sort you guys out later. All you bear-like things, you guys kind of go over there, and I'll sort you guys out later. All you like monkey-like things, you guys kind of go over there, and I'll sort you guys out later. Right, so we started organizing them. And when we did it, we basically created our taxonomic scale, which was actually done by Linnaeus. And so the Linnaean system of taxonomy is something we still use today, but it is basically an organizational tool. So it is us basically organizing all this diversity that we're seeing for really the first time. And we've seen it, right? We just didn't really pay much attention to it. But now we're starting to pay attention to it. We're starting to realize what's out there, and we're starting to organize it and catalog it and all that sort of stuff. Okay. So the Linnaean system of classification. Is basically like a biological 20 questions. Have you ever played 20 questions? Right, it's a pretty basic game, right? I mean, you try to guess who I'm thinking about, and you have 20 questions to do it, right? And generally speaking, what are the nature of those questions? Are they open-ended, short essay questions, multiple choice questions? They're binary, right? It's either or. Are you a male or a female? Well, of course, that one's trinary now, right? So you could say neither, and that would also give you information, right? So are you famous or not? Right? So chances are that one. So you probably didn't want to, you just probably burned a question right there, right? Are you an actor or a singer? It's like, neither. Okay. Are you a politician? Yes or no? Yes. Now we're onto something, right? Okay, let's we can whittle this one down right now. We can cleave these guys in half. Are you a Democrat or Republican? Right, so now, and you can kind of whittle it all down. And so eventually after 20 questions, the idea is you have enough information where you can guess who it is, right? Same thing is also true with the taxonomic system in the sense that you're asking a question about these living organisms and it's binary. What's the first question? First question is this. Are you prokaryote or eukaryote? Whatever that is, right? Well, as it turns out, and this obviously comes after hundreds of years of work. This is not like, you know, at this point, he didn't say like, are you a prokaryote or eukaryote? That domain, by the way, came around 2000. Okay, so the Linnaean system of classification, as we know it now, was not what Linnaeus actually created. Okay, this is a constantly morphing, changing, growing, breathing system. It is a working document. It's just us trying to sort of classify organisms that's useful for us based on what we know now. Every time we learn a little something new, we change the classification system. Okay, and I'm going to talk a little bit about some of the consequences that that has, kind of a bit of a reality check associated with, in particular, evolution, because I want to discuss one particular aspect associated with the thinking of evolution, whereas an example of an oversimplification of evolution. Okay, so what is a prokaryote, eukaryote? So basically, a prokaryote is largely a cell type that is pretty simple. Doesn't have a lot of organization to it. Doesn't have like things like a nucleus and stuff like that. You know, it doesn't have these organelles inside of it where you kind of compartmentalize functions and things of that nature. Bacteria, eukaryotic. Eukaryotes, an example of eukaryotes are plants. Most of what you eat actually are all eukaryotic. More complicated organisms that have compartments to them. They have division of labor in these compartments, these organelles. They have a nucleus, for instance. They're pretty big, actually. And so what we notice is that really you're kind of either one or the other. You're either or. So in the domain, you're either prokaryotic or a eukaryotic. And so our prokaryotic cells, we notice, and this is a later breakup, we notice that there's two domains under the prokaryotic. So these guys are both prokaryotic. So you have domain archaea and you have domain bacteria. So these are both prokaryotic. Archaea is actually the one that forced the shakeup. Like for instance, when I went through college, the domain level didn't exist. There was 5 kingdoms and that was it. It started with kingdoms. So the domain level came later. That's a fairly recent invention. So this actually did not occur until we really started discovering this right here. What's your name? Oh, thank you. So that kind of forced it, right? And where we found those is those deep sea thermal vents. So they're extremophiles. So they live in extreme harsh areas. I mean, areas where we didn't think life could be possible, the archaea, that's where the archaea were. Okay, so they kind of reorganized our imagination of what's possible, what life can possibly do. Fascinating discovery, archaea, but it forced us to sort of reorganize things. And then you have your eukarya, all your eukaryotes. Now, the next question that you ask after you ask the first one and break it up into domains is the kingdom question, right? So the kingdom question is basically taking a look kind of like at sort of like biology plan, right? I mean, is your biology plan the same or is there 4 distinct ones? So there's one distinct way of in which archaea does business. It's very different than the way that bacteria does it. So that's the reason why you know those are in 2 different kingdoms, right? And eukarya, and this is the one I want you to focus on here because we are eukaryotes and we'll go forward unless you're in a microbiology looking at eukaryotes, is you notice you have 4 very distinct ways of doing biology here, right? So you have protists, which is kind of like a unicellular world of eukaryotes. Pond scum, for instance, kelp, they show here, is a type of colonial protist. This is, so if you've ever seen a spongy pond with green slime in it, a lot of protists in there. It's a lot of single-celled eukaryotes in there. Giardia, how many of you guys have danced with that lovely little customer before? You know of it, right? That's a protist. And so that kind of swims around in the water, which is the reason why if we drink the water, it's untreated, then it'll make us sick. Fungi have a completely different way of doing their biology. So do Plantae and Animalia, us. We all have a very different way of doing our biology, right? But you have 4 kingdoms in eukarya: Protista, fungi, Plantae, and Animalia. But you keep asking questions, right? Because you're only just getting started now. So if you just kind of focus on the animals, here's a good example. This is a few questions down. But like here's a big question that sort of separates the animals from each other. Do you have a backbone? Yes or no? The answer is either yes or no, right? If it's yes, then you basically get put into the vertebrate pile. If it's no, you get put into the invertebrate pile, like the insects, for instance, and the soft-bodied animals. We are in, obviously, the vertebrate section, right? If you want to take a look at the vertebrates and taking a look at some of the animals, you could ask yourselves, a question like this, like how do you eat? Are you carnivorous or herbivorous? So do you eat meat? Do you eat others? Or do you eat plants? Well, that's going to split animals into herbivore, carnivore, right? You can continue on, right? The carnivores aren't all the same. You could ask them, it's like, well, are you a mammal or a non-mammal, right? Well, it's like, I'm a mammal. Well, what does a mammal do? Well, a mammal gives live birth and has mammary glands, right? So do you do that? The answer is yes, then you're a mammalia. Right? If you don't do that, if you're an egg layer, like a turtle, then you're a non-mammal. Okay, so that's another split. And so you can see as you kind of start to, you keep asking questions, keep teasing them apart, eventually you're going to get down to the bottom where you have genus and species. And you get something like this. Let's do, how about this one? Anybody know what that is? I can guarantee you some of you guys have this at home. Canis familiaris, it's your pet. It's a domesticated dog, right? How about this one? Yeah, that's a wolf. Hopefully you don't have this one at home. Right, so that's a wolf. Notice some similarities. They both have the same genus name, but they have a different species name. So once you get down to this, by what's called a specific epithet, this kind of two-part name, right? Canis lupus, Canis familiaris, that's what's called your specific epithet. That is like your specific kind of your tag, your ID tag for a species. The idea is there's no other species. These are two different species, right? Canis familiaris is not the same as Canis lupus. Okay, they're considered two different species. But each one of these has a big laundry list of names that go before it, starting with kingdom and then on and so forth, all the way down as you're going through those 20 questions, right? Now, some of you, just to kind of give these folks a little bit of love, have one of these. What is that? Yeah, that's your cat. That's for all you Felix lovers, right? But I mean, a lot of the other cats, so there's different species of cats. So obviously Felix domesticus is the house cat, but you've got other cats like ocelot is also Felix and things like that. And so you have different names, but they're different species, right? So nobody would bring in an ocelot to be a pet. Well, maybe you do if you're in Brazil or something like that, but it's not advised, right? Because I'm just thinking people have tigers as pets and they probably shouldn't. But the idea is, right, this is the house cat and there are different species. Now, notice though, can these two guys hybridize? Yeah, we know they can, right? Because some people created the wolf hybrids. They're a little on the crazy side, but still, right? Now, this happens all the time. And the reason why I point this out is because when you think of something that is two different species, you think of, generally speaking, two organisms that are isolated from each other, right? So, you know, Canis familiaris breeds with Canis familiaris and Canis lupus with Canis lupus, right? And the two species shouldn't hybridize. They shouldn't kind of intermix, right? That's kind of the idea of the whole reproductive isolation concept of species. But here's the problem. And this is kind of where one of these sort of taxonomic sorts of things has direct bearing on sort of the way we frame when we're looking at like an evolutionary study or the change in species populations over time, how this affects those studies. And we need to be very clear and honest about how that's affecting those species or those studies, is because the concept of species is tricky. Generally speaking, we make it very easy for ourselves. We say like, okay, you guys, what is our species name? Homo sapiens, right? Are we a clearly different species than Canis familiaris? Yes, no problem there, right? But when you're starting to talk about the change in species over time, then part of the heart of that discussion, that study, is how are you defining species? And there's like about 6 or 7 different evolutionary definitions for species. And which one you choose will shade your results. So you could have different results if you use a traditional biological view, or if you use a more ecological view or an isolation view. There's different types of definitions and they will give you different results depending on which one you use. Why? Because the concept of speciation is really amorphous. And when we look at like evolution, in particular for evolutionary discussions, we talk about speciation over time or changes in species populations over time, there is an enormous and oftentimes erroneous assumption that the process is clean and neat and tidy, that species A morphs into species B, when in reality, that's not what we see. We see this oftentimes very long protracted transition period where species A kind of starts to sort of mix with species B, and it may go back to A, or it may just fully go and full-fledged into B, but the two will still kind of be able to interact with each other. It's kind of like blending colors, right? So you have white over here, you have red over here, but you have this whole range of pink in the middle as the two are mixing, right? So you have more of a washed out red, you have more of a true pink. So depending on what your ratio is, it's very complicated. And this happens all the time in taxonomy. We're doing this all the time. As a matter of fact, a very good example that you guys probably have in your backyard is an example of what happens in ornithology. This is happening in ornithology all the time. Like when I went to ornithology at UC Davis, I remember learning about one of my favorite birds, which is the rufous-sided towhee. However, shortly after I graduated from UC Davis, the rufous-sided towhee was split. That was a species, right? So the species was the rufous-sided towhee. Beautiful bird. It's a bit of a ground dweller, black on the back. It's got a little sort of rusty red side pattern right here. So it's kind of like got a whole black head. It's got these brilliant red eyes. You probably have seen them actually in your backyard. Beautiful birds, one of my favorites. But what happened was we split them into two different species. The eastern towhee, which is typically from about Colorado, about the Central Flyway, which is just beyond the Kansas border, to the east. And then the spotted towhee, which is the one we have, which is black, but it has white spots on its wings. The eastern towhee doesn't have those white spots. And so now we have the spotted towhee. So now what I learned is the rufous-sided towhee has been split into two species. So one species became two. Now, what happened there? Evolution? No. It was just a reorganization of the Linnaean classification system based on an increase in our knowledge about how these two birds' biology worked. Okay. This happens all the time in ornithology. Lumping, it's called lumping and splitting. Like when we see that two species are actually so closely related that they're probably just the same species, then we lump them together into one species. If we see that the one species is kind of very divergent in the way they look, then we say they're probably two separate species, so we split them. This is happening all the time in ornithology. They're constantly lumping and splitting, lumping and splitting, and lumping and splitting. And it's simply based on how much we learn about them. So we learn something new and we split them. We realize they're actually quite different, or they're not really as different as we thought. So we're going to pull them together. And what that does then, when you're doing that, is imagine you're studying the evolution of the rufous-sided towhee. All of a sudden, by that whole lumping, splitting thing, I've completely upended how I'm going to be looking at how this population is changing over time. Right? So that kind of shades how you interpret things. And so that's an example. That's one of those perfect examples of it's not as easy as you think, right? It's not as simple as you think. To say that it's as simple and shut down as all that is kind of a bit misleading. There's a lot of complexity that goes into this. In a lot of ways, we don't know, for instance, I don't think any evolutionary biologist really knows for sure, is, when do you stop calling it just natural variation? And at what point do you actually call it evolution, right? So when have you evolved into something else as opposed to just naturally like adapted and then you can come back? So nobody knows what that line looks like, to be honest. I mean, a lot of times the problem with that is evolutionary biologists will oftentimes not admit that, but it's a difficult one to find because we just don't have some of the puzzle pieces that we need in order to really figure out how that looks. So we and another thing I always like to do in terms of challenging this idea of life being simple, and that's my biggest beef against a lot of, not the evolutionary biology. If you read what the researchers are doing in evolutionary biology, I mean, they are working hard, they're trying to figure things out, they're doing a great job. It's actually in these guys, the textbook writers. They kind of do a little too much sock puppet with evolution, and they make it sound and come across like a kind of a weak, pasty, shallow. It's not what it actually is. So it doesn't have any of the richness associated with it, which is where my big beef is. Okay. Why? Because life is not simple. It's not. It's super complicated. I mean, it's incredibly complicated. And this is important to know, right? This stuff is not easy. It's not easy at all. I mean, even when we're looking at a process, right? At a process in real time, and we're looking at like a biochemical pathway, it's very difficult to understand clearly and perfectly how that's working. And we're able to generate data on it as it's working in front of us, as it's doing its thing right in front of us. It's still incredibly difficult to really figure out, okay, this is how it's working, because it's just so many facets and so many variables associated with this entire process. So life has all these properties associated with it, makes it very difficult to understand. And that's what we're trying to understand the best we can, what we know now. Okay. Now, the reason why I say that is because life inherently is very complex. It's very well wired, but it's very complicated, and we struggle. The researchers struggle to understand how things are working now in real time. And the reason why I say that is in terms of like keeping your objective perspective check. When we're having challenges and complications to understand how things are working now, it gets even worse when we start to try to study how things were working across millions of years. And I always tell students that, when you're looking at things like, evolutionary ideas and things like this, you have to understand that the things we understand the best are the things that are happening right now in front of us in real time, right? Like biochemistry, cellular biology, things like that, right? Those, because they're happening right here, and we can measure them and we can understand them. And those are the things we understand the best. This is the reason why we have such a robust medical technology industry, right? We've all benefited from modern medicine. Medicine has been built on the back of research, right? If it hasn't been for research, we would not have any of the medications that we enjoy. We would not have any of the therapeutics that we enjoy. All of the benefits of modern medicine all largely came about because of research. That is, researchers trying to figure out how things work in real time. Okay, that's what we know the best. Now, here's the thing: as we start to go back in time, it gets problematic, right? It gets harder and harder and harder to know how things work, especially like from an evolutionary framework. And the reason why I say this is because a lot of times students will notice this, and they'll have like questions or things like this, and they won't understand why exactly. But this is the reason why, right? So they'll take a look at something, an idea that we're trying to put together that was maybe millions of years ago, or hundreds of millions of years ago, and we're trying to figure out how it all went down. Well, here's the problem. When you look back, like the further you go back in history, the harder it gets. It's because it's like a forensic study, right? But it happened so long ago, nobody was there. Nobody was recording it. So we don't really have an idea of how it all went down exactly, right? The best we can do is to come up with hypotheses and potential models that might explain how things went down. And the further back we go, the more problematic that gets, right? So like, for instance, when you take a look at the evolution of the cell, right? How the cells came together, especially eukaryotic cells in particular. We'll talk about that one actually in the cell chapter. I mean, we're talking about something that happened 4.5 billion years ago. So it's almost impossible to know for sure how that all went down exactly. And so as a result, what you see are like competing models, right? There's different versions of the idea. It's like, well, maybe it went down like this. Like, well, it could have, right? Or maybe it went down like this, or that could have as well, right? We don't know for sure because we don't have any real discrete way of discretely, physically testing the idea, because that all happened so far ago, right? And that's just kind of what you see. So usually when a lot of people see those models, they're like, oh, well, that's just a bunch of mythology and stuff like that. It's like, well, science, it's not mythology. It's intelligent mythology, perhaps. But right. But the one thing in science that sets models apart from just like storytelling is that a scientific idea has what's inherently called explanatory power, right? So the idea, like if you have a theory about what happened 4.5 billion years ago, the idea is, even though you can't reach back 4.5 billion years ago and do some sort of an experiment to test your idea, The indirect way of trying to figure out whether or not your idea is valid is to take a look and inspect what's called explanatory power. And you'll hear me use that term. What I mean by that is an idea that has explanatory power is one that has the capacity to explain current phenomena. So you could create an idea and be like, well, if it happened like this, that would explain why we see this and this and this and this today. So the more your idea has the explanatory power to explain current phenomena, typically, generally, not always, but typically, the more robust it is as an idea. Okay, and that's kind of what these models have as they reach far, far back into the distant past, is they have explanatory power. Okay, and so, but obviously, those have those are some soft spots, right? Because you always have different camps of biologists arguing with each other about which way it went down and so forth. And the ideas are intriguing and they're fascinating and they're all possible. And I mean, it could be that, maybe this is the way it happened or this way, or maybe it actually went down some other way. We don't even have any clue yet. We haven't even come up with that idea yet. So maybe it went down some way completely different. And so that's all possible, right? So that's the reason why it's kind of fascinating, is because you can keep working on this, and you see a lot of these camps working on that, right? So that's just something that happens because of the inherent complexity of biology as it exists today, and then it compounds as you try to put it all together in the distant past when nobody was here and there's really no signature that's kind of left behind to help you study things, especially with things like cell evolution, because it's soft tissue, and soft tissue doesn't fossilize, right? So you don't see a bunch of eukaryotic, primordial eukaryotic cells in the fossil record because cells don't fossilize, right? Which actually brings me to my next point, which is really cool. And one of the reasons why a lot of times, and this is one of the big reasons, by the way, why I kind of try to keep that objectivity. I try to say, listen, it is fascinating and it is really cool, right? Like for me personally, I'm like an RNA world person. I kind of feel like maybe RNA was all in place first, not DNA. So that's just because I'm an RNA person by background, because I worked in an RNA lab. But there's a lot of interesting explanatory power issues that it kind of solves that the other model does not, that got hung up on. So there's a lot of things that nature, but this is one of these things. And every now and that, by the way, the idea of an RNA world was really heretical, right? Because mainstream status quo people was like, the DNA molecule came together somehow. Nobody really knew for sure, but it came together somehow. And then it kind of all started from there. And then that all pulled together into a cell. And I mean, there's like any number of problematic issues associated with that entire process, most of which were not resolved in any kind of a significant scientific matter, mostly in a philosophical debate manner, which is not useful at all. But one of the things, because we see this in science, right? We see these what looks like heresy challenges to status quo, which I would argue that kind of the Darwinian gradualist, phyletic gradualism approach to evolution is status quo that needs to be turned upside down. One of the things that is an example of things that sort of make us step back and have to like rethink what we think life was like. Perfect example of this, maintaining our objectivity, is what happened just recently in the last, 5-10 years, which is an amazing discovery. And I'm kind of in the process of trying to read up on what was going on there. But there's a researcher in North Carolina who is recently, this sounds crazy, but she published the paper, and a couple of other labs have also published the paper. She found red blood cells in T-Rex bone. So think about this one for a second. Status quo, phyletic gradualism, all of that, right, says that all those bones in those fossil beds, 65 million years old, they should be what? It should be rock, right? The whole mineralization process, that's how you make a fossil, right? The fossil, the rock minerals will replace the bone minerals. And so, after a certain amount of time, what looks like a bone is actually just rock. It's not unlike kind of what we see with Pompeii, where we see the body cast, where basically there's no body in there anymore. It's just the body was just sort of replaced by the actual the molten rock. I mean, not the molten rock, but the ash cloud, right? The pyroclastic flow. So it's similar sort of an idea, right? So over a certain amount of time, mineralization process should replace all of the natural minerals in the bone itself with rock minerals. So when you dig out a T-Rex fossil, it is a rock that's 65 million years old at youngest, right? Because that's when the Chicxulub happened, right? That's the big crater that destroyed the dinosaurs, right? So here's the problem, though. When you dissolve all that, which she did, I mean, I guess nobody really thought to do this before because they just assumed it was rock. But I mean, she was finding red blood cells in there. Other labs have done similar things, followed up on them, and they found collagen, which is like the main protein in your connective tissue, which to me is fascinating. Because here's a situation where you've got a discovery, according to your status quo thinking, this should not be possible. Because even mineralization, taking its longest to do this, should have long been mineralized long ago. After 65 million years, it's jaw-dropping that it's not just rock. And when you look at that, and I've got questions.
Do you know do they know how it was preserved or something?
No, they have no clue how it was preserved. At this point, they're looking at the mineralization process, but that's problematic because, I mean, the mineralization process is fairly static. I mean, it is what it is. It's like physics, right? So, it's just, yeah, that's why I said it's jaw-dropping. This is one of those moments where you kind of you sort of step back, you stumble back actually in disbelief. It could have been, right? I mean, I think they're right now, they're trying to go through, they're trying to set up all those potential explanations of how did this happen? And what was going on. I mean, it's going to be it's an ongoing discovery. So it's very much kind of in the phase where it's like, oh, I don't believe those are the results you actually got. That's kind of where they are now. So as they kind of pull out of that, they kind of pinch themselves and realize, yes, you are still alive. Right. Then they'll start to move toward the okay, let's go ahead and start hypothesizing, trying to figure out how did this happen, that sort of a thing. That's where they're hopefully they're moving into that. I don't know if they are or not though, but see, and that's the big thing, right? Because then you start looking at this, and so you had her work on T-Rex, you had other labs finding collagen on the other bones. So then you start looking at these big deathbed areas, just like up here that we have just over here in the dinosaur monument and stuff like that, these big graveyards of dinosaur bones and stuff like that. And now you got to start looking at these things and wondering, it's like, well, wait a minute. Is this what am I looking at here? I mean, is this like an outlier? Where just some sort of a weird fluke happened to the process? Or am I looking at a lot of intact bone here? Because that's a game changer. And I would say a really cool one, because where I go, being a molecular. So by the way, my bias, and I'll be honest with you, my bias is not evolution, even though that's what I got my degree in. My bias is genetics. So when I look at anything like this, I think I look at genetics. And when I look at soft tissue and T-Rex bones, which are now bones, obviously, because you wouldn't have that in rock. Now I'm thinking, wait a minute, if you're recovering connective tissue, protein, and red blood cells, mostly protein, DNA is more stable than protein. Are we going to be able to find T-Rex DNA? Seriously, we have cloning capacity already. We can clone. All we need is the DNA. We would at least be able to sequence the T-Rex DNA. We could clone a T-Rex, and we could actually come up with an actual real Jurassic Park. Yeah. Well, I mean, that's assuming we're right. See, that's part of the problem, right? Because when you take a look and you read everything about T-Rex, most of it is storytelling. Most of it is we're guessing. Nobody really knows what T-Rex was like. Nobody. We assume that just because his teeth look terrible, that he ripped up meat. But maybe not. I mean, I don't know. I mean, there's probably a lot of things about the actual organism. I mean, it could be that, we clone a T-Rex and the T-Rex is a pussycat. It's like Dino on the Flintstones. Go watch the Flintstones. No, seriously, go watch the Flintstones. Those are classics. It should be multi-generational, right? I mean, we don't know. That's the reason why one of my favorite scenes in Jurassic Park is when what's his face, the paleontologist, realizes that he's out of a job. But he's like sitting there watching like these two dinosaurs do something, and he has this epiphany, like, we had it all wrong. Yeah, that was such an awesome moment, because that's like so science. Because we get to the place, and we are really bad about this, because oftentimes in science, we oftentimes are the smart kids in the class, right? We're like all the researchers, and we get used to always having the right answers, and we get a little comfortable with what we know, and we get to this really big ego spot where we say, oh, okay, this is the way it works. In reality, that's not really the way it works at all. And that's why I love that scene, because that happens periodically in science, where it kind of gives us a little bit of a gut check. It's like, you know what? Remember back then when you thought you had it all figured out? You weren't even close. You weren't even close. But you know what? Other than to the egomaniac who is in love with their idea, that's an exciting time in science. That's the reason why I love to share this T-Rex story, because it's like, I have no clue what's going on there, but my gosh, that is so cool. I mean, that is like, it gives me chills cool, because that should not be possible. If I were to say at UC Davis, when I was going through there, that someday we're going to find red blood cells and collagen in T-Rex bones, they would have laughed me out of the institution. That's how heretical this is. This is like finding human bones in Precambrian sediment. That should not physically be possible. That's why it's so fascinating, because what that tells you, it should make you excited as a scientist, because you're objective and you want to know what's real. And now for a second, you realize, like, oh, wow, this is so much different than we thought. This is so exciting now, right? Because now you've got this entirely new Disneyland of reality that you can figure out. It's just like, it's like showing up at Disneyland for the first time.
You know her name?
Oh, not off the top of my head, but I will look it up. And if you want, I can post the paper. It's really fascinating. And the other thing also that I like to toss in here, and the reason why I'm a big proponent of maintain your objectivity, don't fall off the idea, is because most of what we know about evolution is antiquated, right? It's 19th century fossil record stuff. And honestly, I've never been all that impressed by paleontologists. I mean, phones and fossil records are just yawn, right? I mean, it's right up there with zoos. It's basically a rock embedded zoo. It's like, yeah, that's great. I'm not convinced. I mean, sorry, not there yet, right? But let me tell you what is fascinating and what's causing evolution to come to grips with itself and sort of change and morph and update, right? From this antiquated 19th century concept to a more updated, accurate, I would argue, in the end, concept is the Human Genome Project. Anybody aware of that one? No, nobody cares. See, as a geneticist, I get all excited. Human genome, everybody's like, painful. So what is the Human Genome Project? We sequenced the entire human genome. And we did it actually a while ago, like more than 20 years ago, actually. Not only that, but we sequenced the genome of a lot of other organisms as well. Matter of fact, we now have the technology where it took us to sequence the human genome, it took us like a decade to do it. We can probably sequence a genome pretty quickly now. Grab something, housefly, let's sequence it, boom, done. So we can sequence the entire genome. Now, what does that mean? So what that means is it opened up the entire field of genomics. Have you ever heard of that one before? Oh, you should. Because genomics is pretty much recalibrating everything. So basically now that you can see all of the DNA sequence in your entire human genome, everything, good, bad, and ugly, all of it, it's kind of forcing us to sort of re-inspect what we think in terms of evolutionary terms that we saw with just like morphology, that is our body shape, right? So it's like if you all have fur or that sort of thing, right? So morphologically, what we built in the 19th century, we are now kind of able to get to this place where we're not just sweep it away, but just kind of like replace it with more of a genomic view, right? Because the idea is this, and this is the reason why I'm a genetics person. Because evolution is a genetics thing, right? The idea of evolution is that transmutation of species, that is the change of species over time. Well, how do species change? Genetically. Because everything you need for making your traits is in your genes. If you want to change the way you look, you change what? Your genes. If you want to change the way a species behaves, you change what? Your genes. So if two organisms are getting substantially different from each other to where they become two separate species, guess what you're changing? Your genes. So rather than looking at evolution as a morphological thing, which leads to a lot of mistakes and errors, by the way, it's a genomic process. And now the world of genomics is finding all sorts of crazy cool stuff that traditional phyletic gradualism has to struggle with and has to answer. And I think if they do so objectively, it will come out as a stronger, more robust concept. Okay, so there's that. So I took a little bit more time with that than I expected. However, I feel like that is necessary. That was necessary. That was a digression, certainly, right? But that was a necessary digression because a couple of things that I'm really sold on is that a lot of times when students encounter science, they oftentimes lead themselves into bad areas because they don't understand how science is wired. But it's also important to understand the sorts of things that, as in science, we are struggling with and we're working with currently, right? So rather than being a stiff, stodgy set of answers in a textbook, it is a living, breathing discipline that's constantly growing. That's the message. And it's growing in all sorts of cool spaces. Okay, so I will start with the big biological questions of life, and we'll start it with that one. Okay.